With more than 6,000 small and micro-cap companies listed, if you're looking for the next apple at the earliest stage, then Channel Check truly is the orchard. The listed companies support Channel Check, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA-licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for Channel Check so you're always up to date on what's going on at the small and microcap data place. Welcome to the C-Suite series presented by Channel Check and Noble Capital Markets. Noble is an SEC-registered, FINRA-licensed broker-dealer and the source of the equity research available on Channel Check. Today's interview features Genprex, NASDAQ ticker symbol GNPX. Noble Senior Research Analyst Robert LeBoyer interviews Genprex President and CEO Rodney Varner. Visit channelcheck.com or click the link in the description for Noble's research on Genprex, as well as news and advanced market data. And now, here's Robert and Rodney.
Hello, I'm Robert LeBoyer, Senior Analyst at Noble Capital. With me today is Rodney Varner, the CEO of Genprex, a company developing a gene therapy platform for cancer and diabetes. Welcome, Mr. Varner. Can I ask you to describe a little bit of what you're doing and the markets that you're serving?
Well, certainly. And first, let me say that I'm very pleased to be here, and thank you for giving me the opportunity. We're developing gene therapies for cancer and diabetes. Our gene therapy program for cancer is initially targeting lung cancer, which is the second most common form of cancer and kills more people than any other type of cancer. The markets for late-stage lung cancer drugs, as we're pursuing, exceeds $20 billion a year. We have strong preclinical data indicating that our gene therapy, REQORSA, is synergistic with two of the largest selling lung cancer drugs, Keytruda by Merck, which is its largest selling drug and had 2020 sales of around $14 billion a year. Also, our data suggests that we can successfully combine with AstraZeneca's Tagrisso, which is AstraZeneca's largest selling drug and had 2020 sales of about $4 billion a year. So that if we're successful in combining with both of these drugs, we would cover a large majority of the late-stage lung cancer market. So extremely large markets and extremely large unmet medical need. Now, on the diabetes front, our diabetes program is preclinical, but if I don't have sales numbers for all the diabetes drugs, but I can tell you that about 10% of the population of the United States has diabetes, and our gene therapy is potentially applicable to both type 1 and type 2 diabetes. So again, we're going after really large markets and really substantial unmet medical needs.
Wow. Okay, and you mentioned that your drug is a gene therapy. Can you describe the genes that it's delivering and what that would do in the patient and for the cancer cells?
Well, let me clearly distinguish our two programs. The first is the oncology program, the gene therapies for oncology that are completely separate from the diabetes program. The oncology program is much more advanced, and so I will address that one first. Our platform is capable of delivering a wide variety of genes to a range of different types of cancer tumors and to be combined with a number of other approved lung cancer drugs. Our lead product candidate, REQORSA, uses the gene TUSC2, TUSC2. It's also sometimes referred to in the literature as FUS1, FUS1. This gene is a tumor suppressor gene it's found to be missing or deficient in around 80% of lung cancers, and also in a range of other types of cancer. What we're doing is using our proprietary delivery system to deliver this therapeutic gene TUS2 into tumors, where it replaces or supplements missing or deficient genes, replacing the tumor suppressor function of those genes. It has a range of different mechanisms of action, and it's referred to by the researchers as a multifactorial therapeutic gene.
Okay, so you're delivering something that suppresses the cancer cells in conjunction with other drugs that will either open them up to the immune system, like Keytruda, or inhibit the signals the way Tagrisso does. Would that be correct?
That is correct. Our drug also showed activity alone in a monotherapy clinical trial, and we believe that it would definitely be worth pursuing as a monotherapy.
Well, given the response rate of something like Keytruda, it works well, but the response rate is low. If you're lucky enough to be one of the patients that responds, you do well, but the majority of patients don't respond. So this sounds like it could be a complementary mechanism of action that could improve the response rates of the checkpoint inhibitors and be used in conjunction with them. Would that be correct?
That is correct. And I think it's also the case that even among the responders to Keytruda, nearly all of them ultimately relapse. They ultimately will become resistant to Keytruda. And that's also the case with Tagrisso. And our data suggest that if you combine our gene therapy with either of those, we can benefit patients, in some patients who wouldn't have responded to one of those drugs. And also, we can extend the time that they benefit from those drugs.
How is Recourses delivered to the patient?
We have a unique proprietary delivery system that we call the Oncoprix delivery platform. We use DOTAP cholesterol nanoparticles. They're non-viral nanoparticles that encapsulate DNA plasmid. These nanoparticles are put into a liquid solution and injected by IV. They circulate throughout the bloodstream and attach themselves to tumors wherever the tumors are found. So they target tumors, and they go into metastatic tumors, wherever they may be found, and also tumors that haven't even been identified yet. I'm sure, as you know, many gene therapies for cancer in the past have used viral delivery systems, which are injected by needle directly into the tumor. And of course, they're limited in that they can't reach tumors that are in places, that can't be reached by a needle. And they also have side effects like generating immune responses that limit their application. So this delivery system is really key to our oncology gene therapy platform.
Sure, that's definitely something that differentiates you from the viral and the AAV approaches, as well as giving potential not only for efficacy, but also for surveillance and metastasis. And could you just describe a little bit about how it's administered, how the administration works, and how many times during the course of therapy with Keytruda or Tagrisso a patient would receive it?
Well, first of all, our therapy alone is delivered. It's the nanoparticles carrying the therapeutic gene are mixed in a liquid solution and delivered by IV. It's a standard IV takes about a half hour. It's similar to what patients receive in countless, other drugs. And so it takes about a half hour. The nanoparticles circulate through the body and attach to tumor cells. Now, in the case of the combination, what we will do is simply add it for patients, for example, who are receiving Tagrisso. When they progress on Tagrisso, we will add our REQORSA gene therapy, and they'll then continue to receive Tagrisso in the normal course and with the addition of our gene therapy. And it's the same with Keytruda. They would just continue their treatment that they've been on and have our gene therapy added to it. Now, these are our current clinical trial populations. This would be second line, what I've just described to you. Of course, we plan eventually to be first line, and in that event, we would be simply added. It would be a combination of the other drug plus us, from the beginning. But again, our upcoming clinical trials are second line, and it will just be added to the drug the patient is already receiving.
Okay, great. And speaking of the clinical trials, what stages are you in, and can you describe the enrollment and what we can expect in terms of data milestones going forward?
First of all, we've done 2 prior clinical trials with REQORSA. We did a phase 1 clinical trial using our drug alone as a monotherapy. That showed a very acceptable safety profile along with strong evidence of efficacy. Then we did a second clinical trial. It was a phase 1/2 clinical trial combining REQORSA with the EGFR inhibitor Tarceva. Tarceva was a first generation EGFR inhibitor. For many years was the dominant selling EGFR inhibitor in the United States. We did a phase 1 trial with Tarceva that again showed very favorable safety and evidence of efficacy. And we started a phase 2 with Tarceva. We have interim data from that phase 2, which shows that strong 7 out of 9 patients who received it got stable disease or better. That is the combination of our drug with Tarceva. So that's a very strong indication of efficacy. There was 1 complete response among those 7. But we know that over the last some years, Tagrisso came along and has now overtaken Tarceva as the largest selling EGFR inhibitor in the United States, and it's the preferred EGFR inhibitor by many oncologists in late-stage cancer patients. So we had originally applied to the FDA for a Fast Track application for the combination of our drug plus Tarceva, the earlier drug. And during the back and forth of the Fast Track process, we were allowed by the FDA to switch our combination to Tagrisso because those are very similar drugs, similar mechanisms of action. And evidently, the FDA must have believed that our data with Tarceva was sufficient to grant the Fast Track with Tagrisso. And I think they recognized, along with everyone else, that Tagrisso was becoming the favored drug. And so we got the Fast Track for the combination of REQORSA plus Tagrisso. And so that's the population we're pursuing in the current clinical trial.
Okay, and speaking of populations, how many patients do you think this would be applicable to out of the total population, and what are those numbers?
These patients who receive Tagrisso inevitably become resistant to it and need another drug. Their alternative right now is chemotherapy. And so our Fast Track is for us to have the opportunity to be an alternative for these late stage patients. And so we can be in the position of treating these patients as they progress on Tagrisso, as they become resistant to Tagrisso. So we have the potential to participate in this multi-billion-dollar market with Tagrisso. And we have the similar situation with Keytruda, which has even larger sales.
In terms of the mechanism, you mentioned Keytruda and large sales. Keytruda is approved in many indications. Would the TUS2 gene and its delivery apply to other tumor types? And is there anything to suggest that it could be expanded beyond lung cancer?
Yes, there certainly is. TUS2 might be effective. It's certainly implicated in a range of other cancers, including breast cancer, triple-negative breast cancer, renal cell carcinoma, glioblastomas, and others, soft tissue carcinomas. And so, I think any of those that might also respond to Keytruda would be candidates for this combination with Keytruda. It's a similar situation with EGFR inhibitors. Any cancer that might respond to an EGFR inhibitor that also has a missing or deficient TUS2 gene would be a candidate for our drug, and there are a lot of them. I think the TUS2 gene is found to be deficient in something like 80% of cancers.
Yeah, you mentioned some large patient populations as well as some small patient populations that are very difficult to treat.
Yes.
So it sounds like it has a lot of potential beyond small cell lung cancer.
Well, it really does. And to clarify, our initial population is non-small cell lung cancer, which I don't know if I misspoke, but I just wanted to clarify that. Non-small is about 80% of the lung cancer market.
Just switching gears to diabetes, could you describe a little bit about the program and what you're doing there?
This is a totally different type of gene therapy. It uses a more traditional AAV delivery system, a viral delivery system. And what they're doing, and this is all ongoing at the University of Pittsburgh, they're using an endoscope to physically deliver the virus carrying the therapeutic genes into the pancreas. And these genes transform alpha cells in the pancreas into beta-like cells. They refer to them as beta-like cells that produce insulin. And that distinction of beta-like cells versus fully beta cells is important because they believe that these beta-like cells are different enough from natural beta cells to avoid the immune system response that creates type 1 diabetes to begin with. Others have tried transplanting beta cells into the pancreas to restore insulin production in type 1 diabetic patients, and with limited success because the immune system destroys the new cells just as it did the old ones. And the researchers at Pittsburgh have found that the immune system doesn't immediately attack these beta-like cells, and they believe that it's because the immune system is not recognizing them as beta cells. So they've seen prolonged responses in mice, which is very encouraging.
Wow, so you're transforming the cells enough to make insulin, but not enough to be recognized and destroyed. That's a really interesting idea. And how much insulin do these cells produce relative to the native production of insulin in unaffected people?
I don't know the answer to that, and I suspect that it may be too early to come up with a uniform percentage like that. I think, I mean, they've definitely seen insulin restored in mice, and they've seen durable reinstating of the insulin production. But I can't give you a percentage of how much is replaced.
For something at this stage, the fact that you can do it at all is quite a breakthrough.
Yeah, I mean, and I think it's important to note that mice were able to live without insulin injections for an extended period of time. So, these were fully diabetic mice, and so they obviously had enough to survive.
Any projection on when this might might start clinical trials?
It's really too early to project that. The researchers did the initial research in mice, which has been published. Last year, they received a $2.6 million NIH grant to continue their research in primates, and which they're now doing. And we're waiting for results from the primate research right now. We know that the researchers are encouraged by what they're seeing, and they're remain very enthusiastic about the program, but we don't have study results from the primates yet. And so what they're doing is the researchers are working to optimize both the delivery system and the delivery vector and the dosing in the primates. And when they have done that and are ready to proceed with toxicology studies, then we will step in and assist them with the toxicology and the other steps necessary to move to a clinical trial. But we're not there yet.
Sure. Okay. It's then it sounds as if the next data will come from the primate studies, and that's what we'll be looking for.
Yes, that's correct.
Very encouraging up till now, at least.
Yes, it is.
Could you talk a little bit about the cash position and the company's financials?
Well, we're in a strong cash position to achieve many important milestones in these clinical trials.
Could you summarize what some of those clinical trials and those data points would be in the next year or so?
Sure. We will be very soon initiating both of our clinical trials, Claim 1 and a Claim 2, those being in Tagrisso and Keytruda, that is combining with Tagrisso and Keytruda. By some people's definition, we've already commenced that Tagrisso trial because we already have IRB approval for it and have a protocol on file for it. But in terms of enrolling the first patient, I can't give you an exact time, but we're optimistic that it'll be soon. So we expect over the coming year we'll be announcing the initiation of those two trials, enrollment of patients, treating of patients, data from the phase 1 portions of those trials from both of them, and potentially other developments from those trials as well. We also have an ongoing research program that's generated exciting results and continues to do so. For example, just a few days ago, we announced preclinical data presented at the AACR conference in both our immunotherapy combination and our Tagrisso combination. That was very important, and we expect that those types of announcements will continue from that program and also from the diabetes program. We're also developing some preclinical assets that we haven't announced yet on our pipeline. They're called in discovery. We think there may be additional announcements from those as well. And then we also are fairly continuously evaluating new technologies, either for in-license or developing internally, like the diabetes program. And we may have announcements from those as well. So I think that we're looking at a really exciting coming year. And we're really excited to have gotten to this point and be facing all of these really exciting activities.
Yeah, this sounds like quite an exciting program and something that is different but complementary to some very big selling drugs for some very large patient populations, as well as patient populations that have nothing and no effective treatment and have very poor prognosis. So this sounds like a very interesting program, and I'm looking forward to seeing those data milestones and following the company.
Well, thank you very much. I really appreciate that, and we're looking forward to it as well. And we're very optimistic and excited about this year.
Okay, well, thank you very much. This has been a very interesting discussion.
Well, thank you. I appreciate the opportunity to speak with you.
Robert, this was a fascinating interview. I have had people in my family who have had cancer, so it's fascinating on how they were attacking this, this problem. But what is also interesting is that they are kind of joining themselves with a very successful drugs already, and it seems like a big market opportunity. How do you look at that?
I see that as synergistic. What they're doing is adding a gene therapy and correcting a gene defect that's common in cancer. They're testing it in combination with Keytruda and Tagrisso, two big multi-billion-dollar drugs that are commonly used in these types of cancers. And in addition to non-small cell lung cancer, these drugs are used in other tumor types. So if it's effective in non-small cell lung cancer, there's potential to expand use into the other tumor types and many additional patient populations, either large tumor types that are common or smaller tumors where there's no effective therapy and prognosis is rather poor.
And everybody knows somebody who has diabetes. It seems like, how novel is their approach to diabetes?
I'd say it is very original, and what they're doing is transforming one type of cell in the pancreas into something not quite a beta cell. It's transformed enough to start producing insulin, but not so much that it would be recognized by the immune system. So it's just enough to give the functional characteristics to the cell to make insulin, but it has potential to be a long-lasting effective therapy. It's early in development, but very interesting. And I've seen the attempts to do things through transplantation or other types of transformation directly, but this is very original.
And Robert, what's interesting is that the drugs that you were mentioning, multi-billion-dollar drug opportunities there, and yet when I look at the stock price, it seems like there's a disconnect here. What are your thoughts about that?
Yes, I agree. This is a very unnoticed, unknown, undervalued situation. And the company is trading at about 150 million or so in market cap with about 56 million in cash, so that its technology value is very understated compared with where they are and the potential for this particular therapy. As a gene therapy, it's in a field that really doesn't attract attention from the large the largest group of investors because of the nature of gene therapy and the early stages that it's in. But the potential here is enormous, and the company is trading at about 3.5 times cash, which is far lower than peers and far lower than other immunotherapy adjuvants that are seeking to improve on the response rate of checkpoint inhibitors or tyrosine kinase inhibitors. So by comparison, this is a deep value, and there are other things about this company that have a lot of potential that I don't think is reflected in the stock price.
Thank you for joining us for this C-Suite interview presentation brought to you by Channel Check. Visit our YouTube channel for more interview content, as well as virtual roadshows and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelcheck.com or click the link in the description to access equity research, news, and advanced market data on this and the 6,000 other small and microcap companies listed.